All right, everyone, we're going to go ahead and get started. Thank you so much for being here, everyone. My name is Cinnamon Moffat. I am the Associate Director of Marine Research stationed here at Hatfield. And we are located in Newport, Oregon, which sounds a little weird for those folks that are here in the room, but this is a hybrid event. So that means we have a whole bunch of folks that have joined us online as well. And I just wanted to say thank you to you all for spending your time with us today. Very excited to have today's speaker. And I think I have a little bit of an echo off of your computer. Do you want to try to mute that for me? We're still playing with technology. Thank you, everybody. Wanted to just go through a couple of logistics and make a couple of announcements.
Not me.
Not you? All right. Do you hear it?
Let's try this.
Okay, that sounds better. Thank you. Sorry, everyone. For those folks that are online, we do have your cameras, mics, and screen shares disabled, but if you put any questions into the chat box, our volunteer, Roseanne, hi, Roseanne, will help navigate any technical issues you have or read out any questions when we get to the question and answer tonight. For folks in the room, I'm looking around, I know most of you, but remember, if you have a question for tonight's speaker, if you just raise your hand, I'll bring you a mic when we get to that place, and that way folks online can hear your questions as well. Also wanted to let you know there is a sign-up sheet on the side here, but that's just if you want to get notifications of these Science on Tap events and you're not already getting them. But again, thank you so much for being here, everyone. I wanted to make one quick announcement. We have our next Science on Tap, which will be November 13th with Dr. Steve Rumrell, who's in the audience. There's Steve. Woo! Steve is going to be talking about shifting near-shore ocean conditions and the consequences of Oregon's marine species of greatest concern. So hopefully you'll come back. It'll be at 6 o'clock, same place, same time, November 13th. So I hope you join us for that. For everybody who's online and everyone in the room, tonight's event is being recorded, so if you'd like to share it with others, it should be posted on our website in a couple of days, hopefully by tomorrow afternoon if I'm good. And then on that same page, you can get information about any of our upcoming events, so if you'd like to join us, you're welcome to do that. But I wanted to start by just welcoming our speaker for today. Dr. Julia Parrish has a lot of accolades, and I promised I wouldn't read them all out. But I am just thrilled that she's here with us. The last time she gave a Science on Tap was actually during the pandemic, and we were fully remote. So I'm really excited to have her here in person. And I'm just going to hand this off to you and get started.
Thanks, Simon. Okay. Oh, good. Thank you so much. So I've been told I can't go outside the blue box. So I'm gonna probably pace like a tiger and get to the edge and then. So my name is Julia Parrish, and I'm a professor at the University of Washington in Seattle, where I have been for a long time. I am in the School of Aquatic and Fishery Sciences and also in the biology department. So I span that solution science, basic science divide. And my day job is actually that I'm the Associate Dean for Academics of the College of the Environment. So I spend my days trying to tell faculty what to do, and they don't listen to me. But I play the long game, so I win in the end. And my nights and weekends job is that I'm the executive director of the Coastal Observation and Seabird Survey Team, or COAST for short. So are there any coasters in the audience? All right, current and former coasters. It's so lovely. Almost everywhere I go within the coast geographic range, which is just south of Mendocino, California, in Elk, California, and all the way up the coast, up past the Arctic Circle to Havoc, the former Barrow, and then out to the end of the Aleutian Islands, the Commander Islands, which are actually owned by Russia. So a really lovely footprint. And whenever I'm there, it's really lovely to give talks in community and have coasters come. So thanks for coming. Okay, this is where I make it work. Okay, so you can't give a scary talk about birds without thinking about the birds, Alfred Hitchcock's The Birds. You know, so this is a really interesting thing. I thought to myself, well, like, that's me when I was younger, running away from the crows. Although Hitchcock's film had featured a lot of crows in it, the book, which is by Daphne du Maurier, if you know her, she was a very prolific sort of scary story writer. The book actually featured gulls. And so it actually was about seabirds, and it got changed because crows, I guess, are scarier. So before I forget, I always forget to do this. I'm just putting this slide up to tell you that the stories that I'm going to tell you tonight and the data that we have collected are because of lots and lots and lots of different funders and thousands and thousands and thousands of people who have gathered the data. So I am super standing on all of their shoulders. Couldn't do it without them. Okay, so this question is a question I often start with, and it's really worth thinking about. Can citizen science be another part of the toolbox of science? So just the way the toolbox of science has microscopes in it and has telescopes in it and now may or may not have AI in it, does it have people in it? Can citizen science be part? And there's two things to think about with respect to citizen science. One is that citizen science should be authentic. It should have real discovery science or solution-based science. So sometimes in formal education, K through 12 or even K through 16, we force our students to do science labs, right? And the labs are sort of predetermined and we know what's going to happen in the lab. So it may be a discovery for the student, but it's not a science discovery. It's not something that we didn't know before. So it's about learning, but it's not about advancing knowledge. But citizen science, I would say, should both be about learning at the individual level, that there's an aha moment, right? But also, ideally, that we move science forward, that we gain knowledge as a collective. So that's one thing about citizen science. And the other thing, and this is this is really a scary story for me. It's free choice, right? So my students in my class at the University of Washington, it's not such a free choice, right? They can leave and they can freely choose to fail the class, right? That's their choice. But somebody who's engaged in a citizen science program, if they're bored or they're frustrated, if it's not working for them, if they're not getting the feedback they need, they leave, right? And so a really good metric or measure of how good your program is how long you retain people in the program. And so that's really something that we think about a lot in designing citizen science programs. What is going to attract people to the program and what's going to keep them engaged or involved in the program over time. Okay, so this is what we tell students. This is the basis of science, right? You make a hypothesis, and then you design an experiment to test that hypothesis, and then you analyze the results and you geek out in statistics land, and then you draw a conclusion. And here's a really cool thing from a science point of view. That conclusion, of course, leads to another hypothesis. And this is a do loop, right, called a career. And you can just go around it for 50 or 60 times, throwing out a few peer-reviewed papers on the way, right? And that's a career. And here's the thing about it. You know, we're not born with a hypothesis in our mind. It's something that we come to after we've been in a place for a while. So the very first thing, especially in the wild, right, outside of the laboratory, outside of the classroom, the very first thing that we have to do is discover a pattern and wonder about what created that pattern in the real world. So some very, very famous patterns in marine science and marine ecology, the striations or the stripes of the intertidal. right? That the mussels are growing in one range, and then the gooseneck barnacles are growing, and then the acorn barnacles are growing, right? And so if you stand in a place where there's a vertical wall, you can see those stripes. Super cool. What creates that pattern? That question led to a very famous career of Bob Payne, who did a lot of really cool stuff in marine ecology. So he didn't start with a hypothesis. He started with an observation. And that's very great, because what that means is that anybody can be part of the science team, because everybody makes observations just by being alive. Okay. Now, unlike science, citizen science often has an action step. So when I talk about science and the do loop of science, and even starting with discover pattern, another way to think about pattern and wondering what created that pattern and going out and investigating that is you're bearing witness. Often you're bearing witness to forces that are changing the environment. And you know this because it's an environment that makes sense to you. It's an environment that you're attracted to. It's a set of places that you go back to all the time. It's your backyard, right? And you understand the pattern in that backyard. For instance, there's cyclic parts of the pattern, right? Summer, winter, right? And that ebb and flow is something usual. You know what to expect, right? You know that the temperature's gonna drop and the rain's gonna fall, right? And it's gonna get dark and things are gonna happen. The plants are gonna stop and then they're gonna start up again. And so not only are you used to that cyclic pattern, but you anticipate it. You make a mental model of what's gonna happen, right? And imagine that if spring started 3 months late, you would think, what's up? What's wrong, right? Why is this not happening? So not only are you aware of patterns, but you keep a model of them in your head. So cyclic patterns, ebb and flow, not only of seasons, but think of tides, are really important in structuring how we think about the world, the outside world, the coastal world, and whether we think it's okay or not. So bearing witness to change is that cyclic change, which actually should make you feel good, because the flowers are coming up at the right time, the salmon are coming back at the right time, the birds are coming back at the right time. But there are two other C's that we talk about in coast. One is catastrophic, and that is when there's a huge big change. And I'm going to talk about some of those tonight. Those are scary stories. But the other one that I just want to leave you thinking about, and I'll get to it again right at the end of the talk, the third C is chronic change. Chronic change is really, really slow, right? So a chronic disease moves very, very, very slowly. And from one day to the next, it's really hard to tell if there's a difference. But maybe 1 week to the next, or 1 month, or 1 year, you can really see a change. And we can do this in the environment as well. So when we talk about a change in climate and that being provoked by greenhouse gases like CO2, but other gases as well, it took a very, very long time for us to get around to, as a community, as a nation, as a world, to really understand that this signal was going up, because it's very slow, but it's inexorable. It's going in one direction and it keeps going in that direction, right? So when you think about chronic change is that thing where when you were young, or you cast your mind back to when you were young and you think, wow, there used to be, the peninsula at Nye Beach used to go out much farther than it does now. And I can remember, right, that it went out at least another half block, and that's gone now, right? That's a chronic change. It was a little bit and a little bit and a little bit and a little bit eroded away. And only after decades do you see that change. So keep those 3 C's in mind: the cyclic change, the catastrophic change, and the chronic change. Okay, so rigorous citizen science. Not only citizen science, but rigorous citizen science. It's 5 things: training, collecting, verifying, analyzing, and reporting out. And you can see that it's a marriage, which means that it's sometimes great and often uncomfortable, between scientists and coastal residents, both of whom are very necessary. So scientists like me, I'm a card-carrying scientist, come and do trainings. I'm doing one here on Friday. And I train people who live in coastal environments who want to go collect data. And I'm going to tell you about the data tonight. But we also verify those data, not so much the basic data, the pieces of data, we call those evidence, but the deduction that people make from that evidence. And in this case, in my case, it's what bird is in front of me. So we analyze all of that out in a big sort of melting pot of analysis, and then we report it out in a whole bunch of ways. We do geeked out peer-reviewed papers, which almost nobody reads. And get this, we have to pay to publish. What is that? My husband is a retired architect. And when we first got together, I remember coming home the first time that I had gotten a paper accepted in a scientific publication since we had been dating. I'm like, honey, right? I got my paper in ecology. And he's like, I've never heard of it. What's ecology? And I'm like, I'm trying to explain, well, it's like a scholarly journal. And right. He's like, well, are there pictures? I'm like, well, no, it's not pictures. But there's a lot of words, and and it's really prestigious, and it only cost me like whatever it cost me at the time, $800. And he's looking at me like, oh, why would you pay money to put just words in a journal that nobody reads? And I'm thinking, that's a question. Yeah. So we report out, we do it in peer-reviewed journals, but we also we give talks like the one that I'm giving tonight. We have a website with lots of interaction on it. And we actually are pretty connected to the news media. So whenever there's a big die-off event, we get lots of interviews. Okay. So the program that I'm talking about is the Coastal Observation and Seabird Survey Team. You gotta have a logo. It has to look cool. Has to be stickers. Stickers are really important. I brought stickers with me. And in Coast, we are doing a standardized search. So the same way, repeated monthly and effort controlled, which means we start at the same place and stop at the same place. So we know that piece of beach is going to be the same piece the whole time. And we collect verifiable data on beach cast birds. Yes, that is to say, this is a program about dead birds. Oh, wait a minute, there it is. I just love this, right? Fortunately, there aren't a lot of vultures near the beaches. We actually do manage to see some beach cast birds, some dead birds before the vultures get to it. So just think about scary stories as totally dependent on your point of view. Okay, so why document dead birds? Why not do live birds, right? Why not go the eBird route? There are millions of people that do eBirds. There's a lot of data. It's great. Why not do that? Why persist in this kind of gross, almost 12-year-old boy thing? Well, depending on where you are, and this is one of the places there actually are quite a few beach cast birds, some more at some times of year than others. And of course, a dead bird was once alive. And okay, so here's the thing. Unlike a live bird, which can be very hard to identify, right? Anybody who started down the path of being a birder, right? And you get to warblers and sparrows, just give up now, right? It's like being a diver, and the first time you go to the reef on a reef dive, and somebody says, let's do the blennies and gobies, right? Tiny little, yeah, right. It's a fish, right? Okay, but a dead bird can be identified by anybody, and that is because they patiently wait for you to find them. So you don't have to get up early in the morning. You do not need specialized gear like high-powered binoculars or a spotting scope. You don't have to wear camo. You don't have to sneak up on them. You can take your coffee with you and your dog. Dogs are very good at finding beach birds. And with the right equipment, like gloves. You can pick them up. You can thoroughly examine them. It is super, super easy to take a picture of a dead bird. In fact, you can arrange them. Brush the sand off, right? Ruck the feathers down nicely. You can put a scale ruler in, right? You can line everything up. I have the world's largest collection of dead bird photographs. We now have over 1 million in our archives, right? So not only am I the dead bird lady, I'm the dead bird lady with a lot of photographs. Okay, and so if we just found 1 or 2, that might tell us something about the health of that bird, but it really wouldn't tell us anything about the ecosystem. Probably wouldn't tell us anything about population dynamics. But when we have hundreds or thousands, or in our case, tens of thousands, we get to tell much larger stories about what's going on in the system. Okay, so here's some basic stats from Coast. We're about 25 years old. We have 400, give or take, sites from Mendocino, that's in Northern California, up to Uktiavik, that's the former Barrow, just above the Arctic Circle. We work in more or less about 90 different coastal communities. And right now we have about 700 active participants, but we've trained over 5,000 people. And we work with sister programs in BC and in Central and Southern California. So all that stretch means that we have more coastline that's monitored for beach birds than anywhere else in the world. And I know that you are sitting there thinking, well, that's probably because you're the only place in the world that does beach birds. And that's actually not true. Coast is one of the newer programs. So looking for beach birds is a time-honored thing, both in the Netherlands and in the UK. So very old, well-established programs there. Also in New Zealand, also in Australia, some fledgling programs in Argentina, and sort of on and off in Peru and Chile. So Coast has found over 100,000 bird carcasses of about 200 different species. Okay, and here's the heart of Coast. This is the Coast science team. Lots and lots and lots and lots of people. It's really fun to have a photo archive of coasters. And I should change this slide up more than I do because it's showing people at a younger age than they are now. But that's nice, right? Isn't that nice to see yourself younger? And what I can say is, of the things that you might think unite coasters, like they all do blank, they're all birders. Absolutely not. Most of them are not birders at all, right? They're all left-leaning environmentalists. Nope, nope, definitely not. They are all young. Nope. In fact, the average age is a little on the old side, but there's a big, wide range. The only thing that we can find that unites coasters is love of place. They have a really strong attachment to the coastal environment and usually to a particular beach or a set of beaches. It's their beach. They speak about it that way, as in my beach. You can't go on my beach without my permission, right? To which I say, okay, your beach. Okay, what's it take to participate in beach birds? Depending on how birdy your beach is and how long it is, it'll take 1 to 5 hours a month to do a survey. In this part of the world where the beaches are very wide, we strongly suggest that people go in pairs or even in teams because it's really hard to search a beach that's much wider than this room by yourself. And also, and quite honestly, science goes so much better when you're with somebody else and you can argue, right? So this Coast is a really, really great plan for any marriage, right? Because you can go out and you can do your survey and you can argue with your partner vociferously, and it's called scientific discussion, right? And so it's great. You can sort of get all of that anxiety out and you can do science at the same time. So 2 things that are happening. One is surveying the beach, and the other one is when you find a carcass, as this couple has, dropping on your knees, snapping on some gloves, and opening a book, which makes other people on the beach think that there's something weird going on, maybe a cult kind of thing going on. And people often stop and say, what are you doing? So it's a really nice engagement, entertainment thing for coasters. So what are the pieces of evidence that coasters are collecting? Well, they're identifying the foot type. It turns out that the number and arrangement of toes tells you a lot about what that bird is, because that's conserved within groups of birds. So birds that are related to each other have very similar feet. We're also taking up to 3 measurements of the bill, of the wing, and of the foot. And in fact, if you can't make any one of those measurements because the carcass is so disarticulated, we say it's not a coast bird. It was a bird, but it's not a coast bird, because coast birds have to have the evidence that we take to prove that it is what it is. And then there are 2 photographs, back shot and belly shot, like the one shown here. And all of that is collected in this very archaic material known as paper. Right. And here's a really fun thing about paper, and you write on it with an article called a pencil, or sometimes a pen, right? Space pens work really well. We use write in the rain paper. The really nice thing about write in the rain paper is when you go out and it's sunny, and then it clouds over, and then a shower comes through and it rains on your write in the rain paper, that's fine. You just shake it off. Now, what do you do when you're taking data on your phone and it starts raining? Do you shake it off? No, you don't, right? And what if you're walking along and you see a beach bird, and in your enthusiasm to get to that bird, you miss the fact that it's in the surf zone very close to the waves. And so you drop to your knees and a wave comes along, gets your boots wet, moves the bird, picks up your data sheet, and takes it back out to sea. Well, you can just run out and get it. You can pick it up out of the ocean and shake it off, right? What happens if you drop your phone in the ocean? So sometimes that rudimentary technology really is good. Also, it's really nice that when we are collecting the data, when coasters are collecting the data, they're giving us photograph shots or screenshots, or they snail mail it in. So we love that we get more snail mail at the University of Washington than any other lab. It's really great. Okay, so there are the 2 photographs, and there's a belly shot. So we like to say that coast is evidence first, the foot type, the measurements, right? Some aspects of the plumage or the color and arrangement of the feathers on the bird, and deduction second. And deduction is that aha moment. What species is it? And how do we do that? Well, you need a key, right? And so in order to be able to use that evidence and say what it is, maybe you know what species it is, maybe it's common, or maybe you're an expert birder, but maybe you're not, and maybe you just need to check in. And so the really important thing for coast and for citizen science is to rip the jargon out of guides. I wish that this was our guide, but it isn't. This is our West Coast guide, Beach Birds, a coast field guide. And I will tell you a very short story. When we were putting this guide together, when we were inventing the guide, we were trying to figure out how it worked, like how you got from lots of birds down to 1 bird. And that's actually kind of, that's hard to do. And it's hard to do so it looks easy. But when we got it together enough, this was quite a few years ago, and my grandmother, who lived to the age of 104, right? She was so mad. Her mother lived to 108. My grandmother was still alive and kicking when we were inventing the Coast Guard. And the postdoc, Todd Hask, who invented it with me, his grandmother was alive too. And so we decided to do the grandma test. And so we Xeroxed all of the pages, mock-ups, and we sent them to our grandmothers with highlighters and a note that said, hey, Grandma, can you just please go through this and just highlight any word or phrase that doesn't make sense to you, right? So my grandmother lived her entire life in East Peoria, Illinois, not exactly a coastal community, right? And she did not graduate from high school. So she's perfect. She's absolutely perfect, right, to check jargon, because jargon for me, I live in science land. So I'm going to use words all the time that somebody else is going to say, what? And they're too polite to tell me that I'm speaking in tongues. But my grandmother tells me what's for or did on a regular basis. So the envelopes came back after a few weeks, embarrassing amount of highlighting. I'm like, is there a single word that they understood? So we had to go through a few rounds. That was the grandma test, and we couldn't pull all of the jargon out, but we pulled a lot of it out. So it was a humbling experience, but a really good one. So here are the things that you need to know to use the guide. You need to make sure it's a bird. And there have been times when people have been uncertain. And then the other thing, of course, and this is a somewhat Monty Python, it has to be dead. It cannot just be resting. All right. How does it work? Well, we start with the feet. And as I mentioned to you, feet are conserved. So we use the foot key, answer some questions, and that gets to a foot type family. The one I'm showing you here is the first one, alcids. They're in alphabetical order. So the first one is alcids. The last one is waterfowl. You follow the arrows, answer some more questions until you get to a box with a stop sign, and that tells you what species pages to go to. So inside of not very many questions, very simple, this or that, you get to species pages. Okay, so here's a sort of rundown, right? Coasters collect the evidence, and they use the field guide to get to an aha. What species is that? They're turning in the evidence and the deduction. We verify all of that. So we have a verifier. You can think of him as the little guy behind the curtain who sits in the dark room, takes the evidence in, makes his own conclusion about what the species was, and then does a comparison. And the cool thing about that is we can then see how good people are. So, how good are people? Well, first, we spend a lot of time asking when people walk into a training, how good do you think you are? Like, if you had to rate yourself in terms of how good you are at identifying birds, how would you rate yourself? And it turns out that only about 15% of people say, I'm an expert birder, or I'm pretty advanced at being able to say which species is which. That's actually not very large. And the rest of that, half of the pie is intermediate, but fully half, just under 50% of people either say that they are beginners in terms of being able to identify birds, or they have no experience at all, which I think doesn't mean that they don't know what a bird is. I think it just means that they are so unsure. They're like, it's a pigeon, maybe, right? So what this tells you is that folks that are walking into a coast training are tentative, right? They're unsure. They think they want to participate. They want to be part of science land, but they're not coming in all cocky about knowing all of the birds. And so we have to train them. Now, the coasters in the audience know this, right? There are some birds that are really hard to tell apart no matter what. So a coaster nightmare is certainly gulls. And in fact, in coast, we take immature gulls that are large, and we put them together in a big multi-species bucket called large immature gulls. And because in birdland, you take all of the species and you make a 4-letter code out of them, a large immature gull is a ligu. And ligus are very, very common on this coast. There's a ligu in the middle right there. This book, by the way, if you don't know this book, A Guide to Troubled Birds, it's fantastic. Fantastic. They make refrigerator magnets out of the art. Okay. So in fact, 5 hours later, post-training, how good are people at identifying birds? We test them. They can get 7 out of 10 carcasses correct to species, right? That's fantastic, considering that most of them were beginners. And they can actually get even better 1 year later, especially on a birdie beach like this one. They can get up to 9 out of 10, right? So again, we know that because we verify. So that's super cool. What that says is that we can take the coast data and put it immediately into science, not only the science that we do, but science that other people do, and we can put it into natural resource management. So that is great because it means the data are used, right? And having your data used is a prerequisite to sticking with the program, right? Nobody wants to go out and survey a beach month after month after month after month and turn their data in and hear nothing, crickets, right? They want to know that the data are used. Okay, so we actually have a lot of science stories. And as I said, we do put them into the peer-reviewed literature. And if you can't live without reading all of that peer-reviewed literature, we're happy to send it to you. I guarantee that if you put it next to your bedstand at night and you wake up a little bit insomniac, you can start one of those papers and it'll just put you right back to sleep. But if you want to read a more storified version, not a dumbed down version, but a de-jargonized version, you can go to the website and look at the science updates. Okay, and this is the Coast website. If you want to know more about Coast, you can go to the Coast website. This is the landing page. If you want to find out more about what's going on in Coast, or you want to join the team, you can go under join our team to trainings and events, or you can just page down the main page until you get to upcoming events, and you can see the talk that I'm giving right now. And I'm giving another one in the same space, much more mathy, tomorrow. And then I'm doing a training here tomorrow, and then another one in Lincoln City. Okay, so I've talked to you all about Coast. I've convinced you it's great data. There are a lot of people involved. We keep them around. It is fantastic. So what? So what do we do with the data, right? So it all comes down to what can Coast do to help science and our environment. Okay, this is the, I think the most important thing that Coast does, the natural history of dead birds, right? So you're used to thinking of natural history being live birds, but I like to think of the natural history of dead birds. Well, one, because I work on dead birds, but also because just think about it. The very last decision that the live bird made was where to be, was where to go, right? Before it got moribund, before it could not fly, before it could not swim, before it was well on its way to death, it made decisions about where to be. And there's a whole bunch of factors, environmental factors, that are pushing birds and other species all around the world, right? Was it the breeding season? Was it the non-breeding season? Is there good food? Is there not enough food? Are there storms? So animals are responding to all of these different things, and birds are no exception. So when we find lots and lots and lots of dead birds on the beach, a decision that a bunch of those birds made when they were live is to be close to the shore. And we know that because when you go out more than about 100 kilometers from shore here in the Pacific Northwest, and you put a bird carcass at the 100-kilometer mark, the chance that it will float to shore is almost zero, right? So the farther out you are, the lower the chances that you get to shore. And we can actually do models of that and map that. So we know that all of the birds that ended up on shore started within a box that's framed by 100 kilometers on the outside. And that's way down to vanishingly small probability. Those birds were probably a lot closer in. So here is a natural history of dead birds. The black line is the long-term average, and the yellow wash is the sort of variability around that average. And the gray bars are what happened in any particular year. And I'm showing you 2016, 2017. And this is all of the beaches in Northern Oregon, which includes here, for all of the years. So the black line is all of the years and all of the beaches. Lots and lots and lots going into that, right? And the gray bars are just that particular year and just that particular month. And you can see that's not a flat line, right? So in come July, August, there's going to be a peak, and there's going to be another one in November. Okay, so just to orient you here, The y-axis is how many birds you would expect to find, how many carcasses you would expect to find in a lineal kilometer of beach, okay? And so you can see that the low points in April, May, about 1 carcass a kilometer, but come August, September, between 4 and 6 carcasses a kilometer. Okay, and that black line again is that long-term normal. So you can see that in this particular year, right, the gray bars pretty much line up with the long-term average. And that makes kind of sense because we use the gray bars to make the long-term average. So this, right, what happened in any given year? This is a pretty normal year. But let's move around in time space and go back a year. Okay, now that long-term average looks like a lot flatter line, but it's actually not. All I've done is stretch up the y-axis, see? So it's not, doesn't stop at 6, now it goes up to 14. So just 1 year previous, 15, 16, during August and September, pretty not normal, right? Way, way more than normal. Let's go back another year. Really not normal, okay? So sometimes we have these wow out events where we're getting thousands and thousands of carcasses. At 32 carcasses per kilometer, which was the signal in December, over all of Northern Oregon, that's about 8,000 carcasses a day coming into Northern Oregon every day for the month, right? That's a lot of birds. Okay, so that's a catastrophe. There's that second C, the catastrophic signal. Okay. So, why? Well, you may have heard of the blob. We used to call it the blob, but then we jargonized it. Now we call it the Northeast Pacific Marine Heat Wave. And then we have to abbreviate that because it's too many words. So when you read the papers, you can't figure out what is going on. Suffice to say that the blob, that big blob of red that got stuck over the Northeast Pacific, right? So think of all of the North Pacific from Japan all the way on over to the Pacific Northwest. That blob of warm water was on our side of the ocean, on the east side of the ocean. And it was up to 4 degrees Celsius warmer than normal, which doesn't seem like a lot, right? So in one day, the air temperature goes way more, way bigger than 4 degrees, right? But it takes a lot of energy to heat up water. And it takes a lot of energy to keep that water warmer. So a 4-degree temperature shift that persists for a long time is an intense amount of energy. And that blob was the size of continental Canada, which is fairly large. And it persisted more or less for about 3 years. The marine heat wave itself was a little less than 3 years, but there was an El Nino on the backside of that that kept the water in our part, in the lower 48 of the blob, pretty warm. So this phenomena, this physical phenomena, changed the ocean in very, very many ways. And the things at the top of the food pyramid, right? The seabirds, the marine mammals, the big, big fish like salmon, pollock, and halibut suffered. Okay, so I'm going to tell you 2 sad animal stories, sad, scary animal stories. One about this guy, Cassin's auklet. It's a bird about the size of my fist. It's very cool. It has electric blue feet, the color that we used to in the 1960s paint our eyelids sort of sparkly blue. Remember that, some of you? I did that. And that's the color of Cassin's auklet's feet. They also have a little white eyebrow. Do you see that on the live bird? That makes it seem like it's always sort of thinking and skeptical about something that you just said. So that's Cassin's. So they belong to the family of birds, the alcids. And all alcids are footballs with wings. So when they fly, they snug their tails and their feet in, and they snug their neck down, so you just see the beak, and then they have wings. And the cool thing is that they can also fly underwater. So these guys, cassins, are small birds, so they eat small things. They eat basically the insects of the sea, including krill, so small shrimp, and zooplankton, and a few larval fish. Okay, so that star is at the northwest tip of Vancouver Island. That's Triangle Island. It's the fifth island out of the Scott Island group. That's the largest colony of cassins in the world. There's about 1.7 to 2 million cassins on that one island. Okay, so that those graphs that I showed you earlier, remember those? When I went through, actually I was going forward and going backwards and showing you the that Cassin's auklet die-off, that big catastrophe. This is what the baseline looks like if we just repeat the baseline signal year after year. It looks like, kind of like an EKG, right? Like you can think of it as a heartbeat graph going ba-bum, 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 ba-bum every year. And you can see that the gray bars pretty much match, and every now and then they don't. And that non-match was Cassin's auklets. Just amazing, amazing. Here in Oregon, we had just done a training when that was starting, and there was a woman who went out, I think it was her second survey, and she found 238 Cassin's auklets, one species, 238 of them in 1 kilometer of coastline. She ran out of material. She called all her friends. She called the hardware store to get more ties that we were using. She called us. She apologized for not being able to process all 238. We said, it's okay. Don't worry. Okay, so what happened? That blob that sat over the ocean did a few things. It changed the prey size, and it also shifted species. So krill in the area went from larger to smaller, and pretty much overnight. And we believe now that part of that was not because they shrunk, but because a new water mass came in with new krill in it. So there was a sort of water mass replacement on a big scale. The other thing that happened is the smaller insect-like things, the copepods, they did shift in species. And we had a loss of the northern species, which tend to be larger and more energy rich, sort of the cliff bar kinds of zooplankton, and they were replaced by southern copepods coming up from California. They tend to be smaller and energy poor, sort of the rice cake kinds of copepods. So we lost cliff bars and we gained rice cakes, and the krill got smaller. And you can imagine, not so good for the birds. The other thing that happened was the only place where there was still a refuge of cold water was right against the coast. And that's because of coastal upwelling. So we had this big blob, but the upwelling phenomena during the summer season held it offshore, held the blob at bay, right? So everything is fine and dandy in that very narrow strip, but only during the summer. So where did all those 2 million Cassin's auklets that are breeding on Triangle Island go? Did they spread out into the North Pacific, which is what they normally do, like these scrappy little birds way out in the middle of the ocean? No, they all came to the coast, right? There are about 5 million of those birds in the world, all of them into that very, very, very narrow strip. And here's the thing about upwelling, it goes away, right? In the fall, when the storms come in like they did today, right, upwelling breaks down. And so the blob rolled in and smashed that upwelling zone. Okay, and that led to the starvation of Cassin's auklets. And because they were so close to shore, we saw them on the beach. So dead birds on the beach are not only about ecology, about what decisions that live birds make, they're also about physical oceanography. Birds as floats and where they end up. This actually caused the death, we think, of about 400,000, give or take, Cassin's auklets in about an 80-day period, which is pretty phenomenal. That wasn't the only thing that the blob was responsible for. This is a larger alcid, a bigger football with wings. It's sort of football sized. common murres. There's more of those than cass and auklets. There's 18 to about 25 million, and there's a lot of jazz hands there because it's really hard to count all of those birds. These guys live a pretty long time, about 20 years. Because they're bigger, they're eating a little farther up on the food chain, so they eat forage fish. You can think of those fish as the size that you would put on pizza, right? So it's about a pizza-sized fish. And unlike cassins who nest underground, murres nest on cliff surfaces. You have some of the most wonderful murre colonies here out at Yaquina Head. And it's really fun to go look at them. Okay, so the blob. Remember I said it was about 3 years long? Well, in the first year, it took out the cassins. In the second year, it took out the murres. So things are coming up, physical impacts, impacting the zooplankton, impacting things lower on the food chain, moving up into the forage fish community. And so the blob did two things. It changed the prey size, so forage fish were smaller, but it also changed the prey availability. Say that word, prey availability. Why? Well, the water was warm, too warm for the fish. So what did the fish do? They went deeper, right, where the water is cooler. So the fish are changing their availability to the birds, right? Because the birds have to dive down into the water. So if the prey are deeper, it's going to take more energy per prey for the birds to forage. Also, at the same time, I love this part of the story. That same warm water heating up the forage fish, they are moving around. It also heated up everything else. So all of the fish are more or less the same temperature as the water they're swimming in. So the big carnivorous fish also, on average, warmed up. And the warmer a fish gets, the higher its metabolism is. And the higher its metabolism is, the more it eats. So one thing that the blob did is make big fish hungrier, make big fish eat more. So imagine who's going to win. In this corner, common murre. In this corner, kind of hangry, hot walleye pollock, right? Murre has to dive deeper, going after those forage fish. Hangry walleye pollock, picking off the forage fish because they're right in front of it because they've gone deep, right? So our estimates are that the large fish, the large predatory fish, won out in that competition, taking relatively more of less prey and outcompeting the birds. And so once again, just like the cassins, murres starved to death. When we originally did the work on this mass mortality event, we jazz hands our way to saying maybe 1 million to 1.5 million birds lost their lives. We didn't know, and that is because the geography over which this event happened was vast, including most of the Gulf of Alaska and some of the Eastern Bering Sea. But we have actually since done some really long-term work on the colonies in Alaska, both in the Gulf of Alaska and in the Eastern Bering Sea. And we now know with fair certainty that about half of the common murres that nested in Alaska died that year, about 4 million. And that makes that single event the largest recorded wildlife die-off ever that we know about anywhere in the world, which is kind of amazing. And definitely a sad animal story. So are there bigger lessons? Coast is just one of the beach bird programs. I mentioned to you that there were other ones. There's British Columbia Beach Bird Survey, BCBBS, and then there is Beach Watch and Beachcombers. All 4 of these programs are interoperable, which means we can all put our data in one big analysis melting pot and look at bigger stories, which we have done. In fact, we combined them together. That gave us 29 years of data, and that's because those programs started before Coast. Coast is the most recent at 25 years, and gave us 90,000 surveys. So that's kind of cool to think about. 90,000 surveys, that's like big data, right? And it's all standing on the shoulders of individual people going out and doing those surveys. So this represents 7,000 or so people. Okay. And we took all that, and what we did is we looked at the incidence of mass mortality events. And so on this sort of railroad track timeline, I'm going to put mass mortality events. Each one's going to be a circle. Big circles are larger, like the common murre event. Small circles are smaller. So they're all fit to each other. So you can see which ones are large and which ones are small. And I'm going to change the colors of them. And the colors are going to say something about which species or which groups of species. So mass mortality events actually have been around for a while. We see them every few years, right? And you can see that they were fairly colorful, but look at that. Suddenly there are more of them, the circles are bigger, and the circles are all in the reddish hue or they're in the bluish hue. And so what we really know here is that when the blob got there, the ocean got stuck on warm, not just in California and Oregon and Washington and British Columbia, but in the Gulf of Alaska, leaping over the Alaska Peninsula into the Eastern Bering Sea. At the very same time that was happening, we had an Arctic warming event, right? Changing the water that is in the Bering Sea and the Chukchi Sea, which is north of the Bering Sea, from a cold stanza to a warm stanza. So we had multiple physical events which were all making the water warmer. Okay, so we can take all of those mass mortality events and we can put it on this one. This is my one geeked out graph. So if you're not a scientist, work with me on this. We can do it together. Yeah. And each one of those red dots is a big one. It's a big mass mortality event. The gray dots are very, very small ones. In fact, small enough that I didn't even show you them on that railroad track. I just showed you the big ones. Okay, so on the x-axis, the horizontal axis is sea surface temperature, and it's anomaly, difference from long-term normal. So we set normal at 0, which means that if it's above 0, warmer than normal, towards the red, and if it's below 0, it's colder than normal, towards the blue, right? So that's that slider. Now, what is on the y-axis, the vertical axis? Event magnitude. What's magnitude? Well, it's a jazz hands kind of concept, and it's got a few things in it. It's got carcasses per kilometer, right? How many carcasses are you going to find in a kilometer of beach? It's got the spatial extent. How many kilometers was the mass mortality event? Was it just here in Newport, Oregon, maybe 5 or 10 kilometers? Was it 100 kilometers? Was it 1,000 kilometers, right? That event scale can really change how many carcasses you would expect to find. And then the third component is duration. How long did it last? Was it just a month? Was it 2 months? The murre event in the Gulf of Alaska, common murres came in at 1,000 times normal for 9 months. 1,000 times normal for 9 months. That tells you a few things. One is normal is very low, which is true. But 2 is, wow, that's a huge event, which it was. Okay, and so what can you see here? What is the relationship? I haven't drawn a straight line. I've drawn a kind of squiggly line. And I used some statistics to draw that. So I'm basically using a routine to say, what's the best line that I can draw to kind of approximate these dots? And that's what that line is. And that line is something that we call a step function. And you can see where you step up in that line, right? And it's just above 0. So above long-term normal to about ½ a degree, we get a step up in the magnitude. Now, let's go back to magnitude. Notice that on that axis, it's a log axis, which means each major step is times 10. So it goes from 1 to 1,000 over that axis. That's a big change. So let's translate this out of science speak and into English. When the ocean temperature goes up 1 degree C for 6 months or more persistently, the ecosystem is going to lose millions of birds. It's very simple. It's also a scary story, right? What do we do about that? Okay. There's some other things that changed in the warming waters. Bull kelp disappeared. And that happened here, definitely happened in California. We had a mass wasting disease of sea stars, and sunflower stars went nearly extinct. And some people ask, are jellyfish taking over as a consequence of a warmer ocean? Well, maybe. Not jellyfish like the one in the movie. I really want to watch that movie. It looks like a sort of inflated clown kind of thing gone rogue. We have seen this guy, Velella by the wind sailor, also associated with warmer water. And that's the importance of serendipity. And so this is a turn the table story. So yes, we are really seriously concerned about all of those dead birds and what's going on and the change in the ecosystem. But we are also seeing other species that seem to be doing well, and Velella is one of them. Now, we're not a jellyfish program, Coast isn't, but it turns out that coasters are curious people. They observe their environment. Remember, we started in that place. See a pattern and wonder about it. So lots of coasters write down on their cover sheet, hey, we saw a lot of by the wind sailor. Hey, there are a lot of Velella. And they take pictures and they send them in. So one year, one spring, we had some coasters here in Oregon call us up and say, hey, you know, we normally see Velella about this time and we're not seeing any. What's up? What's your data say? And we thought, great question. We never thought about that question. So we interrogated our data set and we went in and we used some routines to pull out all of the photographs and all of the mentions of jellyfish or gelatinous zooplankton or blobby things, right? And then we looked at all of those and we sorted out the ones that were Velella. And it turns out that we have more data on Velella than anybody in the world. That's kind of cool. And so this is a time space map of the occurrence of Velella, and blue is no Velella. That's where we have surveys and nobody saw any Velella or reported any. Gray is no surveys at all. And red is Velella. And what you can see is that in that same blobby period, 2014, 2015, there's a red stripe, and it goes all the way down the coast, 900 kilometers, right? Over 900 kilometers of coast, Velella turns up on the beach the same week. Right? It's not like it starts in the north and comes down to the south. What happens is it blooms offshore, and there is very subtle, but happens every year, wind reversal in the spring, so that the surface water, just the very surface, comes ashore for things that are floating just above the surface, like basketballs and Velella. So Velella comes to shore every spring if they're in the offshore environment. And you can see big stretches of time, like 2006 through about 2013, where no Velella came ashore. Is that because we didn't have that onshore wind? Nope. Happens every year. It's a standard part of the year. We just didn't have Velella offshore. So what is the other thing that we needed to get Velella? It turns out what we needed was higher than average sea surface temperature. So we have a hypothesis that what happens is Velella comes into our environment, the California current, it's hanging out more than 100 kilometers offshore, growing up. It's actually a colonial animal, so it's growing up and getting bigger during the winter. And if it's a really cold winter and a really rough winter, those colonies will disintegrate. They never have a chance to get really big. But if it's a warm winter and a mild winter, they do. And then we see them onshore. The year 14-15, where we got a big die-off of Velella, we actually did another jazz hands biomass calculation, and we calculated between 5 and 7 blue whales worth of Velella came ashore, right? Think how many of those little guys go into making one blue whale. Lots. Okay. So I've been nattering on at you for a whole hour. And so I think I want to end here rather than go on, but I want to just give you a few take-home messages for Mischief Night and Halloween. What's happening in the coastal environment and actually in the North Pacific is a pretty scary story. It's a pretty Halloween kind of story. But it's a story in which there are winners and losers. We know that the alcids, including Cassins auklets and common murres, are part of the losing team. They seem to be a family that persistently loses. And we think that that's because they eat close to the edge. That is, it takes them a lot of energy to dive down and get their prey. Also, in the north, there's a second family, the procellariids, the tube noses. Albatrosses are in that family. So are storm petrels breed around here. They also tend to die in droves in persistently warm water. We think that one of the reasons for that, with two of the big parts of that family, northern fulmars and shearwaters, is because they fly a very, very, very long distance during the non-breeding season. And that takes a lot of energy too. So if they can't tank up, if there's not enough food and they migrate, they just run out of gas. They don't quite make it. So yes, there are losers, but there also seem to be winners. So that even though coasters tear their hair out about gulls, gulls seem to be doing okay. And so do cormorants. And so do some other species of birds. So when we see a change in climate, we know that the ecosystem is changing and we will get loss of some species, but we will get growth of others. It's not about an ocean full of nothing, but it is about an ocean full of difference. And if you worry about that difference, you should speak out about it. Thanks for coming to listen to me.
Thank you so very, very much. I know that we're pushing the edge of our normal time, so if anybody needs to leave the 2 doors in the back, you're welcome to sneak out that way. But I would like to open us up for some questions, and I'm going to bounce back and forth between folks online and folks in the room. And I can see Roseanne has a question already primed for us. So go ahead, Roseanne.
How evenly distributed are your coastal observers? Are they pretty evenly distributed? Or do you have pockets of coastline with no observers?
Super great question. So how evenly distributed are the coast sites? In the lower 48, they're actually fairly even, and they are what we call oversampled, which means if you took a piece of coastline, we tend to divide the coastline in about 100-kilometer lengths, and you looked at all of the sites within it, and you took some out, and you calculated the average of carcasses per kilometer with those sites removed, you get the same value as if you put them in. So we tend to oversample in the lower 48 in almost all places. There's a few places in the northern coast, outer coast of Washington that are difficult to sample. Once you get up into Alaska, different story. And that's because there's far more coastline in Alaska than there are people. So those sites are fairly clustered around. And what that actually means, which I think might be the intent of the questioner, what that means is that we're not going to get all of the signals. We're not going to get a mass mortality signal unless it's large in terms of its magnitude, right? So carcasses per kilometer up, but the spatial extent very broad and likely the duration more than a month. Super great question.
All right, questions in the room. Steve, start with you.
Thanks, Julia, for a wonderful presentation and also just for the delivery. Enjoyed that very much.
Sure.
My nerdy question for you is the practical one we spoke about before. How long a baseline should we use to assess kind of the normal pattern? And how can we assess anthropogenic human-caused sources of mortality against these larger, also human-caused catastrophes?
That's a super great question. It's almost an existential question, isn't it? And it's not just a question about beach birds, it's a question about everything. I mean, think about the climatology, right? So if we think about sea surface temperature, that's a measure that we use all the time in coast. And when you ask, well, what's normal? The definition of normal is a 30-year average, right? And we choose a 30-year range of time and we move through it and past it, right? And we're testing against that 30-year average. And then we actually update it and we move it forward. So think about what that does, right? Because the climate is warming slowly, every time we move that climatology forwards, right, it's going to be different than it was in the last period. And so what the new normal is, literally, is warmer than the old normal. And so that's hard to think about. When we really started to look at mass mortality events, what we actually do is take those catastrophic events out of the baseline signal altogether. And that's because they really pull it in particular places. So we have a statistical way of saying if it's bigger than X, and X happens to be 5 times larger than the peak mortality of all beaches for that month in the area. But you know, when we got to 2014-15 and it's like popcorn going off, right? Do we take out half the signal? Is that a responsible thing to do? So now we actually do it both ways. We take out big mass mortality events when we're calculating the long-term baseline, but we also calculate the baseline with it in to see what that difference, what that variability is. The other part of your question is how many years do you need? Well, like any place, it's about the variability of the signal. So in places like that, like Oregon, you have a very, very consistent signal and you don't actually need a lot of years. You wouldn't want to have wonky only years, but with 10 years or even less than 10 years, you can get a pretty reliable signal. If you go to Puget Sound, you need about 25 years. And that's because the signal is very, very, very low. So even a small change makes it very, very, very noisy. And we see a bit of the same thing in Alaska. There are places where the signal's almost zero. Southeast is a really good example of that. The Gulf has much more of that cyclic or seasonal signal. So I think it's like anything, you know, it's a numbers game. Yeah.
Other questions in the room? Okay, coming at your way. And again, if you need to step out, go ahead and use those back doors.
I'd like to echo the speech or talk. Wonderful. Sorry. I can't speak, but you did a great job. So I guess going back to like the MER situation and like ecology 101, do you imagine or predict that these die-offs will cause niche shifts, which will impact forage fish availability, or these mass die-offs affecting all species pretty like consistently across?
Super great question, right? I mean, I kind of wish I was a young ecologist again, because I'm thinking I need another 30 years to figure this out. You know, so here's the really interesting thing. When we see a mass mortality event and we're recording it on the beach, we already know that those birds made the decision as live birds to come close to shore, right? So we've at least figured that part out. But the other weird thing, and it's a really weird thing about that signal, is it's so pointed in taxonomy, right? It's not that you whack off a portion of the whole top of the food pyramid and everybody suffers. It really isn't like that. And so I have at least started to really stop thinking about trophic levels and start thinking about the webbiness, the connectedness of the food web. And I really think about shifts in energy transfer, right? So if each species is a little circle or a node in a network diagram, and the links or the lines between them are the flow of energy from prey to predator, what I think is that there are shifts that are provoked by bottom-up change, by physics, that cause valves to turn off and leave some nodes high and dry with almost no links to them at all. Why that happens to alcids and some procellareids and not other species, I'm not smart enough to know, but I would hazard a guess it's because species who don't live so close to the energy edge of food gathering are buffered, and B, species that have a much wider diet are going to suffer less. Yeah, but it's super, super fascinating why some die and others don't. And it makes me wonder if things go on, right? Is it going to extirpate those species or really smash them down? One thing about the Murre story, I told you that we lost half the Murres in Alaska. The thing that's most depressing and distressing about that story is not that, because in fact, they could have bounced back. So if you whack off half a population in a pretty rich place, but then you return the place to normal, so it's as productive, the Gulf of Alaska is productive as it was pre-heat wave. What should happen to that population? Well, competition's gone way down, right? Because half your neighbors are missing. And there's a lot of places to breed because half your neighbors are missing. So the population should have dialed up, but it didn't. There's been no recovery in the Murres. They've stayed flat for the last 7 years, 8 years since the heat wave. So there's something going on in the system that when things get whacked off to that degree, they're having trouble coming back. That said, common Murres in the lower 48 are fine. Right? So in this part of the species range, they appear to be fine. Gulf of Alaska, Eastern Bering Sea, not so much. Hard to know why. Ooze, tough question over there. I know you're going to ask a tough question.
Are actually going to wrap us up for this
Okay.
evening and let you off the hook a little bit from the tough questions that I know are going to come. I just want to say that if you were interested in this kind of topic of shifting baselines, come back to Steve's talk, because I have a funny feeling we're going to talk a little bit more about that. If you would like to geek out with us and talk more about the data side of things, Julia alluded to the fact that we're going to have a seminar here tomorrow at 3:30. And my understanding is we're going to get more into that data side of the story a little bit more. So you're welcome to come back and join us there. For folks online, thank you so much for being here. For folks in the room, thank you so very much. If you have follow-up questions, maybe you can come up and snag Julia if you need it. But again, thank you everyone for being here. And Julia, thank you so much for sharing with us. I appreciate it.